things hook up about hooking one above it. And that'd be 3 hooking to 5. Like I say, you build it 3 hooking to 5. And on any particular one of them, like right here, there's your 3 end, which is the flat side, and your 5 side is the pointed side. So when you hook them up, this one, you have it pointing down to make it go right, correctly. These are the replicating strands. Okay, this is your DNA molecule. This strand's making a copy, and this strand's making a copy. These binding proteins are just to assist that these things don't get all wrapped up like, hairs. This strand has no problem hooking one to another because that will give you your proper progression. The lagging strand is going to be a problem because you've got to build this way and then build some more and then build some more. And I'll tell you about that more in a minute. As you know, polymerase glues these things together. As a new nucleotide is brought in, they're welded together by polymerase. And you always hook the 3 end of the old nucleotide to the 5 end of the new one, 3, 5. So when the new one comes in, it's the 5 end on him, that's the point. And I told you earlier, it looks like the new ones are always poking the old ones. That will give you that 3 to 5 hookup. It does that. Now this is the picture here. And these are lining up in this direction. Okay? And this is the leading strand because they hook up as they're appropriately hooked up. And if you see here, this is your 5 end, like that. That's your 3 end. And then slide right in and poke. As if your previous nucleotide, you pokes that one as it hooks up, like I told you. Now, this is the parallel strand, that's the anti-parallel strand. In other words, this thing, as it unzips, he just keeps on putting new ones in there. But this one can't do it that way. So you have the anti-parallel strand that we call the lagging strand. Those sugars are arranged backwards. They don't lend themselves to just hooking up one to another. You have to overlap. Okay? If you didn't, then the new nucleotide would have to connect to the 5 end of the old one. And like I said, that just won't happen. It doesn't happen. So, the way it works is that it does a little bit, does a little bit more, does a little bit more, does a little bit more. Instead of going bam, 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 like leading does, the back end, it does some, and it moves ahead and does some more, and it does some more. That little piece I'm showing you that moves, and that's called a Okazaki fragment. The little part that moves is the Okazaki fragment. And the way it works is that the new nucleotides, this is the end that's coming in, that's the new end. 3 is always the new end, 5 is the old end of what you already have down there. Now, a picture's going to come and show you this because to me a picture is better than all these words together. Then Okazaki fragments are laid down, and it doesn't matter. Now it's laid down by other DNA. You know, I'll wait to share my picture. The picture I can explain it better. Okay. Now, when the Okazaki fragment's down there, then the polymerase can weld it to the one that's been there, and it's like welding and welding. This is your welder. Okay. All right. Now here's how it works. Now look at these on the leading strand. They build in a natural progression. It's going to be 3 to 5, no problem. But here, I got to do a little strip at a time. Open up more, do another little strip, open some more, do another little strip, open some more, and I am always kind of that. Instead of walking along sowing my seeds, I sow some seeds, and I run back over here, and I sow some more, and I run back over here. That's how the lagging strand works. And this little segment here that you're sowing, that's the Okazaki fragment. And what happens, the assembly is like a, what do you call the houses that come pre-assembled, you just put together? There's a name for that. What am I trying to say?
What?
You can buy houses with
prefab?
Yeah, prefab. See, right here is prefab. Here's prefab, and then it is inserted. I'll prefab some more and insert it. Prefab some more and insert it. These don't need prefabbing. I just let them fall in the right order. The leading strand is easy to do. But that lagging strand, because of the way they got it, you got to always hook up 3 going to 5. So I unzip some more and 3 going to 5 again. Unzip some more and 3 going to 5 again. While this thing's going bam, 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 bam. No problem. This sucker's doing like little leapfrogs. That's the way the lagging strand works. And as you notice here, this is your primer. This is one that starts it all off. And now you can hook 5 to 3, 5 to 3, like you're supposed to. So here's your Okazaki. Okay. This will come in, and because these are the complementary bases, they attach. Now I unzip some more. I do another Okazaki. I unzip some more and another Okazaki. So these fragments, they're not used with the leading end. That's just bam, bam. That's just no problem. But they're used with the lagging or the anti-parallel end. And I found a PowerPoint. I found a picture on YouTube showing it. It's pretty cool. As it proceeds, the 3 ends of the Okazaki will come and meet each other, and then right here is a placeholder, and that's going to be replaced with the real DNA molecule. This is RNA right here.
The green.
And RNA is used to bring this thing in, and when it gets located there, he'll fall off, and DNA comes up to make the appropriate base. So I said right here, the primer fragments, they're kicked out. See, there's your primer fragment. He's just on there to start to show. And this is RNA. Well, it could be DNA. The main thing, he is not part of the sequence. He's just like the caboose. So he's going to be removed, and then a real DNA fragment comes in, and then it's glued together. It says right here that this primer is kicked out, replaced by the real nucleotide, and then the train is complete. Now, the Okazaki, they're stitched together to each other. They're hooked to each other by it's like a ligament that hooks bones together. Well, the word ligase is a play on ligament to strap together. So ligase will take one fragment Okazaki and actually hook it to the next fragment. So here are 4 Okazaki fragments. That's where they're not yet hooked together. This has been replaced. This has been replaced. This is the real DNA. It used to be the primer. That's the real. Now all left is to glue these suckers together. And that's where ligase comes in. not the other one, not polymerase. Polymerase works down here, it works in here. But to hook this section to this section requires something called ligase. So ligase only works on the lagging strand. You don't need it down here. But they're hooking up just one after another beautifully. And when you're done, this with the ligase becomes that. And you amaze yourself. Well, you're halfway done. I make one down here too, you know. I'm making one down here too. So I'll have the parent DNA, by splitting and providing the opposing parts, will actually produce two brand new DNA. This is the ligase in there, inserted. That's the welder. See here, there's nothing there yet. That's how it's going to look. There's the welder inside there. And when you're totally done, if you look carefully, this thing was built 3 to 5, and he was built. This perfectly because this 3 accepted the new 5. This 3, the new 5. But in this case, they had to go backwards to make the thing work. I found one YouTube where the person was teaching, he thought he was teaching, he just showed both strands and showed them coming in. And a lot of reviewers said, man, that's wrong. That's wrong. You got the leading strand right, but that lagging strand doesn't do it that way too. And they ate his lunch. It's still on there, so somebody saw that one and thought that was the real deal. So you can't always trust everything you read on the internet. I can find I will, but man, folks who are in the know, they ate his lunch. Because David said, we're the Okazaki fragments. Because all he showed was there's T, here comes A, there's C, here comes T. He did it both sides that way. And on this side, it should be going, you know, bam, 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 bam. And he wasn't doing that. And when I was looking for a good good animation, I saw that one. I went, that ain't right. And then I read the reviews, and oh, they ate his lunch. Somewhere I'm gonna have that thing. Let me look ahead real fast. See if I got that. I gotta find it. I don't know where I put it. So when you are totally finished, I have 2 brand new, and these are identical. These are identical. These 2 are just like the one that led to them. So this strand can go into one brand new cell, and this strand goes to another brand new cell. Where you had one cell with this one. Now you got 2 cells with the same DNA. That's mitosis. That's how it's supposed to work. Human DNA is real long. And you'll see it unzip. Like I think I told somebody already this. It unzips several times throughout the length of it. And so it's happening all up and down it. So if I had to wait one at a time, it'd take forever. But if you all help, then it goes faster. So these are called, they're multiplication plate. I don't think places. That's where it's happening. But you know, finally they're going to come apart. When you get done, there'll be 2 total ones facing us. This is how they run together. I mean, he's working. Before long, these join, and you have this. And if you look carefully, this is your original DNA. That's your new DNA. And half of it was what you created from the half you already had. That's that semi-conservative type of deal. But I tell you what, if this is right, that's got to be right. And if this is right, that's got to be right. So all the red indicates is that this was the new addition to make this a complete DNA molecule. So there's your original strands, and they separate. The new ones come in, and they keep on separating, and when you get done, you have 2 brand new, identical, and that's replication. Here's another way of looking at it, and this one I created. I hope it makes sense to you. Here's your original right here. When I get done, both of these had better be the same as that one was. And if you look carefully, they are the same. But I got to split these bonds, and I do that. And as I split them, they come in and produce, and these new ones are put in here. And when you get done, you're going to have two identical strands that both look just like that one did. That's replication. Now here's another look at it, and let's say this is chromosome X, and you see how it twists and turns. It has to be unzipped. And then when you're unzipping, each side is making the brand new one. When you get done, I have two produced, which are just like the one I had to start with. This and this are the same thing. I didn't make this anyway. I made. I think that was took a while to make it, but I did. Making PowerPoint hard part is remember where you're going, that you get where you want to be at. Because sometimes you take off one direction, end up on Mars. You go, that ain't what I want to show them. Now I want to show you. I thought I had it on here, and if I don't, I'm going to go get it.